Hello everyone, I am François Paugam, and today, along with Elizabeth DuPre, we will present you the second part of the data release presentation. Last week, Julie Boyle, Basile Pinsard, and Valentina Borghethani talked to you about the acquisition, the pre-processing, and the activation maps of the data. Today, I will talk to you about functional connectivity on the resting state data, and Elizabeth will talk to you about intercorrelation on the movie 10 dataset. But first, I want to thank the team, and in particular the scanning team and our participants, who worked very hard so that we can acquire such large amount of quality data. And also all of our sponsors, in particular the Fondation Courtois, thanks to whom it was possible for us to undertake such an ambitious project. First, I will talk to you about functional connectivity in resting state data. Functional connectivity in the brain can be seen through seed-based connectivity maps. The idea of that is that even if you're in resting state, so resting state means that you don't sleep but you don't have any stimulus, so you just lie in the scanner and do nothing basically. But even if you do nothing, the activity in your brain is not completely random. To see that, you can choose a seed in your brain, so that's a voxel, a specific point in your brain, and you plot the activation of this seed across time. And you can compare this pattern of activation to all the other voxels in your brain, all of the other places. And what you will see is that some places, they are highly correlated to the place you're looking at. So here the red dots represent the voxels that are highly correlated to our seed. So you see that it's not just random noise here. We have some places that act together, that some kind of coordinated, functionally coordinated. And when you aggregate the voxels that act together, you find that you have networks, functional networks, and those networks are actually quite reproducible through different people and different runs. So that's what the graph on the right is showing, is that even if you redo this with different data, you will get approximately the same regions that will appear on your maps. And so the x-axis is time, and you see that the more data you have, the longer is your time course, and the easier it is to reproduce the same maps. You can see that even with as much as 30 minutes of data, which is not that much, at least in our project is not that much at all, we can achieve highly reproducible results around 0.8 correlations. That's like very good reproducibility. So the second way of analyzing your functional connectivity is actually quite similar. It's to plot a connectome. So the connectome is this matrix in the middle, and what you can see is that it represents the correlation of each pair of voxels. So you number your voxel from 1 to n, and each element of your matrix is the correlation between the voxel I to the voxel j. So actually, if you take one column or one line of your connectome, it actually corresponds to the seed-based connectivity map of that voxel that correspond to the number of the row or the number of the column. But since we have a lot of voxels, so around 200,000, having a 200,000 by 200,000 matrix is not very easy to process and visualize. So what we do is we first parcellate the brain. So we aggregate the voxels in different regions, and we look at the connectivity between the different regions. The data I use for this analysis comes from the HCP-TRT dataset. And in particular, from that dataset, I only took the resting state data. So for each of our 6 participants, we have 5 runs of resting state. Each run is 15 minutes, so it makes for 600 volumes. And all of that combined makes for 1 hour and 15 minutes of data, so 3,000 volumes. I split the data in 2 repetition sets. Each one of them is 2 or 3 runs. So the first repetition set is 2 runs, so 30 minutes, 1,200 volumes, and the second one is 45 minutes, 1800 volumes. And that's nice because you see that the smaller set is 30 minutes. But as I told you earlier on this graph, we can see that the 30 minutes mark, even with our smaller repetition set, we can hope to reach stable reproducibility. Also, as I told you, for the connectomes, we need to aggregate our voxels in parsers. So for that, I use the Ukrainian Atlas. So it represents 7 networks. And those networks actually correspond to the kind of networks you can see from seed-based connectivity. In particular, on top, so the brown one is the default mode network, the same I showed you in the previous slide. But so 7 is not a lot of parsers, so we divided that to 33 regions. So to divide, we actually take the connected parts of the atlas. So that's what you see. The difference is that in the same network, if 2 regions are not directly connected, they are taken as 2 different regions in our 33 regions. So let's look at some results. Here are the seed-based connectivity maps we get for the default mode. The big plots here are the mean across our 6 subjects. Above me are the examples of the 2 repetitions of our subject 3. And next to me is the mean obtained on the nylon fMRI development dataset, which contains 115 subjects. So this one is to give you an idea of what we are supposed to get. This is what looks like when we look at one specific subject, and finally on the other side, the mean of our subject. So you see that all these different plots look very similar, so that's very reassuring for us. So our signal looks like what it should look like, so that's good. So if we compare 2 different subjects, so here are the 2 repetitions of subjects 1, and here just 1 repetition on subject 3. And you see that the difference becomes a little bit higher between subjects, but still the main regions are still the same to be found. So that's still good. And finally, here in this big slide, you can see all the results on our subjects. So with the 2 references, still the mean of our subject and the mean on the nylon data. So you can see that the pattern is still observable everywhere. So that's great. But of course, it's impossible to ignore the outlier here, namely subject 2. So it seems that we have 1 subject that doesn't correspond to the rest. So should we be afraid? Well, actually not that much, because yes, there is a more connectivity here all around the brain, but if we look at the places where the connectivity is the highest, it still corresponds to the pattern we observe in every other brains. To convince you of that, let's change the threshold just on this specific subject 2. And we can see that keeping just the highest connectivity, we still end up seeing the same pattern. And actually, the threshold used can be quite misleading, because depending on the amount of noise in your data, it can artificially correlate all your brain more if you have more noise. And we know that our subject 2 is the one with the most movement, so we can safely assume that this higher connectivity comes from the movements that introduce more noise. But the overall pattern is quite reproducible, so that's good. We can look at a different network. So here's a visual network, and you can see that, like, what we are supposed to see here, this region activated, we can see that it's activated in all of our subjects, and it's like very beautiful on the mean on our subjects. But you can see that depending on the subject, the region get bigger or smaller, and we get attention areas that light up or not. But again, tweaking the threshold could allow us to see that in any case, it's the same region that gets mostly activated. So still good reproducibility here. So let's look now at the connectomes I talked to you about. So here for subject 1 are the connectomes on the 2 repetitions. And so what you can expect from a connectome is to see some kind of block structures, because since we know that the connectivity is scattered around different networks, we should see those networks appear as blocks in the connectome. So here the network of the different regions are showed as the colors on the rows and columns. And we don't see like clear blocks of like different colors, but actually we can still see some kind of structure of different regions that get activated together. So, and more importantly, the pattern is very similar between the 2 repetitions. And we see that the pattern is a bit clearer in the second repetition, that are 15 minutes more data. We still see that like the cleaner connectome comes from the repetition with more data, which is reassuring. So here from another subject, so we can see here with our subject 3, who is the subject with lowest movement and best SNR, we can see that the colors are better separated across the columns and lines, so that shows us that the pattern differentiating the different networks is more present here. And we can better see the block structure around the diagonal of our connectomes. Those are 2 very beautiful connectomes. And if we look at the connectomes of all our participants, we can see that although the structure seems a bit different from one participant to another, every one of them displays this kind of blocky pattern, with maybe the notable exception of subject 2 that has higher overall correlation. But as I mentioned it earlier, this can be explained by the higher amount of movement that introduce higher movement of noise that increases the overall correlation between all the Fox cells in the brain. And now for a more quantitative assessment of the reproducibility of those results. So you can see here the green boxes represent the intra-subject correlation. So for all the different seeds and seed-based maps, and for the connectomes. And in the beige is the intersubject correlation. So as we can expect, of course, the intra-subject correlation is way higher because it's easier to reproduce within the same participant. And actually, we have like quite excellent reproducibility, above 0.8 correlation in intra-subject. And for intersubject, we have acceptable reproducibility, like medium well reproducibility between 0.5 and 0.6. And with the exception of the connectome, which keeps fairly high reproducibility. So I hope that from this graph, I can convince you that our data is of good quality. At least we observe what we are supposed to observe here, namely the reproducibility of connectivity maps. And to examine more in detail the correlation between the participants, now Elizabeth will examine the movie 10 dataset and the intersubject correlation.
Hi everyone, my name is Elizabeth DuPre. I'm a PhD candidate at McGill University working with Dr. Jean-Baptiste Pauline, and today I'm happy to tell you about some of the initial analyses we've been doing with the courtois.neuromod dataset, in particular with the movie 10 subdataset. So one of the first questions we had when looking at movie 10 was how successfully are our movies evoking a shared response across participants? That is, are we seeing very similar patterns of activity when each participant looks at the same movie? One measure that's really common in the literature to use to ask this kind of question is intersubject correlation. What intersubject correlation is a measure of the similarity of bold responses across participants. And one reason it's so useful for these kinds of analyses, for these kinds of questions, is that we've already seen in the literature that engaging stimuli generate higher intersubject correlation values and across more of the brain than unconstrained or ambiguous stimuli. So, for example, here I've reproduced a figure from van der Waal and colleagues where they looked at three different classes of stimuli. Rest, which Francois just told you a little bit about in the connectivity analyses. Inscapes, which is an ambiguous stimuli created by van der Waal and colleagues. And Oceans, which is the film Ocean's 11, a popular Hollywood film. And so if we look first at resting state, you can see that there are very low or even no visible intersubject correlation values across the brain. And this makes some degree of sense because we know that in resting state, each participant is having their own idiosyncratic experience. And so we wouldn't necessarily expect there to be shared signal across participants. Meanwhile, if we look at a Hollywood film like Ocean's 11, we can see that there's a lot of shared signal across participants, which suggests that they're really all experiencing the film in a similar sort of way, experiencing it perhaps as the creators intended. It's very engaging. And if we look at Inscapes, which is this ambiguous stimuli created by van der Waal and colleagues, which lies somewhere in between the two extremes, we can see that, in fact, Inkscapes has perhaps the average between the two amount of intersubject correlation. So it has some strong intersubject correlation values, but not distributed across the entire brain. And so this gives us reason to think that if we look at intersubject correlation in movie 10, we'll be able to see how well our different movie 10 stimuli are eliciting shared responses across participants, how engaged they were in the movies. So this sounds great as a measure, but how are we actually going to calculate it? To use that, we're going to use Brainiac, which is a toolbox put out by the Princeton Neuroscience Institute. And in particular, we're going to calculate temporal ISC. So what that is laid out really nicely in this figure from Nastasi and colleagues. And so you can imagine taking a single voxel in one participant and correlating it with another voxel in another participant. And so each voxel has its own time course, and effectively what you're trying to do is decompose what they call C of T, the consistent part of the time course across viewers, the idiosyncratic part of the time course across viewers, and the spontaneous part of the time course across viewers. And so by correlating each voxel in one participant with every other voxel in another given participant, you can start to get a hint of this if you repeat it across all participant pairs. So what we're actually going to do here is do leave one subject out intersubject correlation. And what's nice about this is it generates a single time course per subject. So how that works in a data set like courtois.neuromod is that we take a single participant at a time, we hold them out, and then the 5 remaining participants, we average all of their time courses together. So we have a single time course for the group. And now we can calculate intersubject correlation between our held up participant and the group time course. And what's nice about this is it quite well approximates C of T, that is the consistent part of the signal across viewers. And in this way, it also acts as a form of denoising in that by better approximating C of T, we're discarding more of this spontaneous signal, what they call S of T. And what's nice about that is the spontaneous signal is often things that we're not interested in. So this is not the idiosyncratic signal. This is often things like motion, for example. And so by doing this intersubject correlation, this leave one out intersubject correlation, we can get a better sense of what the evoked activity for a given movie is on average across our participants. So to do this, we needed the movie 10 data itself. So perhaps you've already heard the first dataset release talk, in which case you're very familiar with movie 10, in which if you haven't had the pleasure of doing that, I would highly encourage you to go check it out. But once we had the movie 10 dataset and we had the pre-processed version through fMRI prep, which is available as part of the courtois.neuromod derivatives, we did a few additional post-processing steps. So each movie was trimmed to a similar length, and this is perhaps most important because the movies are of slightly different lengths, and we didn't want to confound the effect of a given movie stimulus with the length of that movie. So we trimmed all movies to be about the same size, and this indeed follows in recommendations from other colleagues in the literature, such as Aliko and colleagues. We then, once all the movies were the same lengths, did some pretty standardized post-processing, including standardizing and detrending and smoothing with a 6mm Gaussian kernel in order to, again, make sure all our data was comparable for our analyses and prepped for our analyses. So after all of that, what do we find? So I'm just going to show you here first maps for a single subject. This is subject 1, and for each of the different kinds of films in movie 10. So first for Born Supremacy. This is the same map, but it's shown in a volumetric view and in a surface view on top and bottom, respectively. And what I really want to highlight for you is that even though I've thresholded these maps, if I didn't threshold them, it would be very hard to interpret them because it's the whole brain. But even though I've thresholded these maps, you can see right away that we're seeing very, very high values in regions that we would expect. So you see very high values in visual cortex and in auditory cortex. And again, that comes through here. And this makes a lot of sense because these are audio-visual films. So we do expect to see consistent audio and visual activity across participants as they're engaging in these films. We also do see some more frontal regions, which shows that perhaps higher order cognitive or emotional processes are also shared across participants as they're engaging in this action film of the Born Supremacy. If we look at hidden figures, you can see even slightly more widespread activation, particularly in frontal regions. So again, we have this shared high activity in auditory and visual cortex, and again, replicated on the surface, all for subject 1. And again, even though we've thresholded these at 0.1, you can see just how much of the brain is showing a really synchronous response across participants. So now what's impressive compared to Born even is that we see more frontal regions engaged, which makes more sense, which makes some sense, excuse me, since this is a more social stimulus as well in terms of the narrative. Then if we look at life, you can see just as before, we're still seeing very synchronous audio activity and visual activity as well, but now we're seeing less frontal activity. And this, as a documentary style film, may have some reason to suspect why we're not seeing as much synchronous activity in frontal regions as some of the more social films. Finally, for Wolf of Wall Street, we're also seeing, as before, what we expect, very synchronous activity in visual and auditory cortex. And we are seeing more frontal activity again, which you would have to ask the participants what their own experiences of the film were, but would suggest that they were highly engaged in the film at least. Now, what we can actually do is we can look across movies. So here what I'm doing is I'm plotting the different movies, and I'm actually including both repetitions of the life film here. And what you can see in these white squares is I've just drawn squares around where each film is. So every row is a different participant. And then these are the intersubject correlation maps. So this correlation matrix of the intersubject correlation maps shows these sort of communities around the diagonal. What that really means is that each film shows the most similar activity to itself as compared to other films. So you can see that really clearly here, for example, in Life versus Wolf of Wall Street, where it seems like the intersubject correlation patterns for Life are much more similar to themselves than they are to the Wolf of Wall Street. So all this is doing is lightly suggesting that intersubject correlation maps are perhaps relatively stable across participants within a given film, and they are slightly different between different films, which is really exciting because, as we just saw, even though there are some regions which we expect to be really consistent, like visual and auditory areas, having differences between films is also a really exciting thing to explore. Then one last check we wanted to do is to see just what is the average intersubject correlation across all of the different film types in movie 10. So here again, I've arbitrarily thresholded it at 0.1. And you can see that we are seeing very high auditory values, we're seeing very high visual values, and we are seeing scattered shared activation patterns across frontal cortex as well. This is a really promising map because again, even though I thresholded it just so that you can see some definition of the brain and see these higher values, we are seeing a significant portion of the brain that is showing very reasonable ISC. And in fact, if we look at a reference data set in the literature, so this is a figure adapted from Nastasi and colleagues when they recently introduced the narratives data set, which is a auditory data set of many stories. Here you can see that they've done a very similar thresholding at 0.1, but as expected, since their stories are all auditory, you're really seeing a concentration of shared activation along auditory or cortex in the temporal lobe, and you're seeing very low shared activation around visual cortex, but an audiovisual film like the films in movie 10 are able to see more reasonable intersubject correlation values here. So this just gives us really good sense of the kinds of data we're able to see in movie 10 and how it compares to other reference data sets in the literature, suggesting that the data quality in movie 10 is very high and makes it a very useful data set for looking more in depth at these questions of how does shared activity or evoked activity differ between movies and how is it shared in some ways.
Thank you for your attention. I hope you enjoyed this talk. Just before we begin the Q&A of this session, I just want to mention that if you're interested in this project or in the dataset, you can find more information either on our website, on our documentation website. If you want to check the code we use for our analysis, you can check our GitHub. If you want to check the other talks, and in particular the talks on the previous week on the part 1 of the data release, which contains more information on how to actually get the data and begin working with it yourself, you can check our YouTube channel. And if you want a more daily update on what we're up to, just follow us on Twitter. See you in a minute. Bye!